dendrites. They're not here. What you can see are the sensory neuron cell bodies, and this is those types of neurons. So it's this lighter pink inside, the circles, and then surrounding the circles, you might see some darker dots. The darker dots are a type of neuroglial cell called satellite cells. Okay, so the cell body of the sensory neuron is here. That's all you can see. And then also some neuroglial cells as the darker dots. So give me two ways or two terms that I could classify this type of neuron. Sensory and structurally unipolar. Yep, that's what you would do. Let's see what this one is. And that one's good too. So the circle is the cell body. Inside is the nucleus, and then the dark dot is the nucleolus, which again, I don't know if you know. And then here you can see the satellite or neuroglial cells even better. You can just say neuroglial cells, the dark dots. Questions on that? No, they are not. They are not. Satellite cells are in the central nervous system. The cell body of a motor neuron will be in the central nervous system, but they don't cover them the way that they do these. That's one part of what you have to do. Now let's look at the actual model of a multipolar neuron. So for that, I'm going to go to web images. Of course, you can always Google things. Google neuron model images, and you'll get a bunch pop up. So here's where we are for the third practical. This is your section, nervous system. Of course, your practice test, some slides are there, and then diagrams is what I'm going to get into to see pictures of the model. Okay, we're going to start with the neuron. And that's what you have at the tables. Now, the one back there on this side is an older model. Looks a little bit different than this one. They both work. You might see either on a practical. However, I can tell you honestly, usually if I'm setting it up, I tend to choose one of these two. I think they're a little bit nicer. Meaning, at some point before you guys leave, you might want to go check out one of the other ones. Okay. And of course, maybe some of you guys should go check out theirs, because you never knew. All right, ready? If you are sitting at a front table, I want you to go to a back table. And you can pull up your chair or whatever, or stand around. I want everybody to look at the model. And go find the model. I'm going to point the things on the board, and then I'll be around to help. Wherever there's an open space, you can go. All right, ready? We're going to find the dendrites first. The dendrites are these cream-colored, thick extensions here. For you guys, they're the thick gray extensions, the thick ones, not the thin wire. And for you two, it's not the orange stuff. I'm going to come back and tell you what the orange things on top of them are. But it's just the thick cream-colored extensions. Everybody see that? Those are dendrites, receiving signals. This whole big thing is the cell body. Inside you see the nucleus. And around the nucleus, do you see the blue lines? For you guys, it's blue stuff all over the place. And for you guys, the other two models, they have red dots on them. Okay, that blue with the red dots, or in your case, just the blue stuff, those are Nissl bodies. Nissl bodies. N-I-S-S-L, and all of these terms should be in bold in your lab manuals. Nissl bodies, you also need to know the function. They produce proteins, and that includes neurotransmitters. Neurotransmitters. And neurotransmitters help conduct a synapse or help conduct an impulse from one cell to another cell.
So produce proteins and neurotransmitters.
Yeah, the neurotransmitters are a protein.
Right.
Yeah, so if I'm just being general, I could say proteins. If I want to be more specific, it would include neurotransmitters, which are super important for conducting the impulse. And I know we haven't talked much about it yet. We'll talk more about it hopefully on Thursday. Okay, now, go down from the cell body and find that thickened area. It looks like a funnel or a triangle. That is the axon hillock. Here, it will be determined if or if we will not conduct an impulse. If we do conduct an impulse, if it's enough, and I'll explain how that we figure that out later. But if it's enough to produce an impulse, then and only then will it go down this red line, which is the axon. So if you look at this area, they cut it so you could see inside. So the red line is the axon, and just the red line. Is your guys is red too, right?
Yeah.
It's red? Okay. Now, do you see on the tips, here and here, where it's not cut? So this is the axon that's not been cut. That whitish covering, for you guys,
It's orange
I
or
forget
pink.
what color.
Pink.
It's pink? Okay. That is the axolemma. The axolemma is the plasma membrane of the axon. And that's a term in your lab manual in bold.
Where?
It's not on the.
I don't know what page.
It's in bold on 134.
134?
But it's
I'm
not
hearing
on the
134.
picture.
Axolemma. Plasma membrane of the axon. And again, it's the white, or in their case, the pink layer around the red axon. Okay, now, go look at the yellow. You see everything in yellow? Everything in yellow is myelin sheath. The myelin sheath is made up of lipids, fats. It helps to insulate the axon, and it also creates a faster conduction speed. Now, this cell is, I can tell by the setup of the myelin sheath, just by looking at that model, that this is in the peripheral nervous system and not in the central nervous system. Does anybody know how? You do? Huh?
It has nodes around here.
Yes, and what creates the nodes of Ranvier? Why are there gaps between?
Schwann cell.
Yes, exactly, good. Because I take one look at this, and I see, you see the red dot on the outermost layer of the yellow myelin sheath? It's like a reddish dot, kind of covered by the yellow. That bump, there's one here, and there's one here.
Those are the.
For you, they should still look like red bumps, right? Kind of stick out. Okay, those are the nucleus, specifically, of the Schwann cell. So, the Schwann cell, remember, when it makes myelin, it's like its arms come around and fold and keep folding and folding. That ends up becoming myelin. So the myelin is part of the Schwann cell. So the Schwann cell goes from here. Let me draw on this. It goes from here to here. That's one Schwann cell, and there's the nucleus. Here's a nucleus. This means it's another Schwann cell, goes from here to here. And yes, because of that, that is why we know that this is not in the central nervous system. Because these cells are Schwann cells, short segments, you need a whole bunch on one axon. That's Schwann cell, that's peripheral nervous system. If this were in the central nervous system, you wouldn't see the cell body on here at all. Because remember, that was an oligodendrocyte that had one, left my stuff in my office. Okay, draw. That had, the oligodendrocyte was here. and then it had foot extensions that covered sections of different axons. Well, if the nucleus is right on the myelin sheath, that has to be a Schwann cell. So you can't be in the central nervous system. So yeah, just by looking at that, that's how I figured out that this must be a motor neuron and not an interneuron. Because interneurons are also multipolar shaped. The only thing that's going to look different is how the myelin sheath is set up. Okay. Next structure are the nodes. Can you find the nodes? They are the little indentations between the two different Schwann cells. You see, it's not like my pictures. My pictures, you could see it nice where it was an actual opening that wasn't covered. Here in the model, it doesn't show you that very nicely. So all you get are these divots.
Can we just call it node? Or we have to write it?
Well, I would like you to say nodes of Ranvier for full credit. Can you just say nodes and have people know what you're talking about? Yes. But since we're trying to get all the scientific terms, I'm going to ask you to say the whole thing. And then the final thing, do you see this bit of gray tissue here at the base? Oh, that's not the final thing, I'm sorry. But at the end here, that gray tissue is called the endoneurium. See that mold? Endoneurium. Now, this is a connective tissue covering. It's not nervous tissue. It's connective tissue. But it covers individual axons. It covers individual axons. Now, in real life, this gray tissue would cover this whole thing. They cut it off and removed it so you could see what's underneath it. But in real life, it would be gray the whole way up the axon. It would cover the whole, on top of the myelin sheath. It would cover the myelin sheath. Now, when you say a nerve is more than just one axon. It's a whole bunch of them together. So what you are learning about now is one of 3 tissue coverings, connective tissue. The endoneurium covers individual, one axon in its myelin sheath. But then you're going to have another middle layer and then an outer layer that covers and wraps around an entire nerve, which I'll tell you about later. But this is similar to, what did you guys do with this? Let's see, we're going to do this with muscle cells, where there's an endo, a peri, and an epimysium. Didn't you guys do something? Oh, well, it was endosteum, periosteum. Okay. Well, anyway, you're going to see these similar terms continue as you go throughout learning about AMP. Now, what you are seeing is not the end of this neuron, because they just cut it off abruptly. In real life, this thing would continue until it went poof, made its teledendria, and then each one would have a synaptic knob at the end of it. Let me draw that. Okay, do you see what I'm saying? This is not the real end. In real life, this is not the end of an axon. So that axon underneath the myelin sheath and endoneurium would continue, and then it would go poof, poof, poof into those teledendria, which are the small branches of the axon, and then each teledendria ends in its own thick bulb called the synaptic knobs, which are also known as axon terminals, synaptic bulbs. I say synaptic knobs, and they've also just changed the lab manual to also say synaptic knobs. They used to say axon terminals. Anyway, it doesn't matter which one you use. Okay, do you see what I mean? So they cut it off. There's only a couple of structures we're missing, and they were back here, and they were on the dendrites. So for these 2 tables, that orange thing that's sticking on the dendrites for you guys. It's the little wires, those gray wires that end in that gray bulb, little gray thicker area. Okay. Everybody see those? They are coming from a totally different neuron. They do not belong to this neuron. No.
Wait, those are the synaptic bulbs and the teledendria from the
Yes, from a different neuron. Exactly. So here's a neuron, another neuron here. It has its dendrites. It got the signal, cell body, axon, sends it away, sends it away, sends it away, and then it goes poof, teledendria. All of these are teledendria that end in the Hershey-Kiss shaped structure, the more bulb-shaped structure. That's the synaptic knob. But do you see what I mean? It's from a totally different neuron. They're trying to show you how one neuron communicates with another neuron. And that would continue down here, and then when we get to the synaptic knob, just like there, we'd send it to another neuron. And keep going until we got to the muscle cell or the gland or the heart, wherever it is we're going. Okay, so the top part is a portion of the teledendria. You can no longer see the axon from this neuron. That's cut off. All you can see is the ends of its teledendria, and then the thicker Hershey-Kiss shaped bulb, the synaptic knob.
So the end of each teledendria neuron are attached, like that's the way they communicate, like the end of that one attached to the end of
Okay, so the end of one teledendria and then its synaptic knob connects with the dendrites of the next neuron.
the dendrite, not
Yeah.
not the teledendria or the synaptic.
No, they don't go end to end. It's
Right.
like one tail to a head, and then a tail to a head, a tail to a head. Not the two teledendrian synaptic knobs. You never have two synaptic knobs together. It's a good question. What's the difference? There's no difference. It's just it's showing you parts of two different neurons here.
Oh no, she said the difference between the names. Synaptic. Synaptic is this.
Which is the teledendria?
Yes.
Okay, put your hand on the thinner part that you can move all around. That's teledendria. Now go down to where it touches the dendrite, where it's thick. That is the synaptic knob.
Is this structure macroscopic or microscopic?
Oh, this is all microscopic. Yeah, so let me show you. Okay, so here would be the teledendria, and then the synaptic knob like this. So the thin part was the teledendria, and then the thick part at the bottom was the synaptic knob. Now, between the synaptic knob of that one neuron and the teledendria of this neuron, that area, so let's say this is the, well, first of all, I have to say, technically, they don't actually touch. You'll learn about that in lecture. Well, actually, you're going to do some of that in lab, too, on Thursday, when we do neurophysiology.
Is that where the synaptic gap
The
is?
synaptic cleft, yes. The opening space is the synaptic cleft. And then what happens, the communication between one neuron, the synaptic knob, and the other neuron, that's a synapse. So when one neuron sends a signal to another neuron or another cell, that is a synapse. And they do that by using neurotransmitters because they don't actually, in real life, they don't touch like it showed you on your models. They don't technically touch each other. So they send these chemicals across. But we'll learn about that on Thursday. Okay, I'm going to, we have more stuff to do, but I'm going to pause there a moment, and I'm going to give 2 little lectures or whatever, where I'm going to have, you can gather around me at the tables. But I'm going to do it twice, so, however, spread yourself out. Okay, grab that model, and I'm going to grab this model, and I'll start the first group, whoever wants to come over to that table. I'm going to show you the stuff on both models in case you get both. Well, I did tell you I usually prefer but, I can still show you the same stuff over here. So if they have it, I can do it too. Doesn't matter. Sometimes we just have preferences as instructors of which ones we like. I happen to like this one the best, but you just don't know. No? It does look dangerous. All right. How is everybody gathering?
Looks like something you put at the end of the training.
Well, I can do it one time. Can everybody see, or do you want me to do it twice?
Do it twice.
I'll do it twice. Okay. I'll do it twice. But if you're here now, I'm going to ask you to step back so the people who are not up here will get back. We'll have first few later.
Yeah.
Okay, just warning you. All right.
So after this, we're going?
I'm going to do it again, but I'm saying the people who are sitting will have first row. If you want to gather behind them, you may. All right, your first row this time. So remember that these are multipolar neurons and motor neurons. Functions, major parts: dendrites. Dendrites. You see the signal. This big area is the cell body. The nucleus. Now, in this one, it's the blue tubes, because it's like endoplasmic reticulum, if you remember that. And it has the red dots on it, which would simulate ribosomes. So this is called the nisobodies, and it's where proteins, including neurotransmitters, are made. Now, the nisobodies here are the blue things everywhere. They're all these blue little pebble specks.
You say they synthesize ribosome?
The ribosomes make proteins. So they include the red dot ribosome structures. But what they do specifically is make neurotransmitters as the protein. Yes, nisobody. Okay, so that's the blue. And then you come down to this thickened area. This is the axon hillock. And then the red line is the axon. Okay, red line is the axon. It's cut in half so you can see inside of it. Otherwise, if it was not cut, you would not be able to see the axon, because actually on the outside, if you notice in this one, it's white. Like right here, it's white. That's the axolemma. Here, it's still kind of a pink color. That's the axolemma. If we didn't cut this, again, all you would see would be the axolemma the whole way across. You got to cut it to actually see the axon, which is it's the middle portion. Okay, axon, axolemma. Now all of the yellow is the myelin sheath, and in this case also this outside pink. That's specifically called a neurolemma, which you don't have to know in lab, but you do in lecture. It's just the outer layer of myelin sheath. Again, it's lipid structure, fat structure, helps to insulate the axon and speeds up the nerve impulse conduction rate. If you look at each segment of myelin sheath, you will see this red projection sticking up, red bump, kind of embedded in the myelin sheath, the outer myelin sheath layer. That is actually the nucleus of the Schwann cell. So this whole area is a Schwann cell. This is another Schwann cell. They each have their own nucleus. One Schwann cell and then another Schwann cell. So they make and keep wrapping around to produce the myelin sheath. In between the two Schwann cells, where it divots in, like here's better view, this indented area, those are nodes of Ranvier. And this is where the nerve impulse jumps, but you'll learn about that later on. This gray area is the connective tissue covering. It's not nervous tissue, it's connective tissue covering that is called the endoneurium. Endoneurium would actually go the whole way up and cover the entire axon. It would cover all of the axon and the myelin sheath. You wouldn't even see any of this if the whole thing was intact. Okay?
So it covers it to this point? Or?
Yeah, it covers it to about this point. Yep, where the axon begins. Now, we've got this. Remember that they cut this off. In real life, this would have continued until it reached its end, and it would poof, branch out, teledendria, and have its own synaptic knobs. Okay, we can't see that here. Instead, we have dendrite cell body of a different axon, or different neuron sends its axon, poof. And all of these thinner branches are the teledendria of that different neuron that we can't see.
Not one different one, but individual different ones. Like this is the
Oh,
different-
possibly, yeah.
Okay.
Yeah, this might be all from one neuron. This could be from 6 neurons. Yeah, that's,
So
you know, we can't tell.
right. Basically,
it's
you
cut.
just have an end. It will be.
It would look like this, yes. It would look, this would have been at the end.
But you said they're not really connected.
Here, these two areas? No, okay, no. In real life, these are not connected. There's a space between and they attach either to the dendrite or the cell body. Most to the dendrite, though. But yeah, there would be an open space, and we'll learn more about that on Thursday. But this part is teledendria, the thin part, and then where it's bigger, like a bulb, that is the synaptic knob itself, like a little Hershey kiss at the bottom. So where this neuron synaptic knob comes together with a dendrite of a totally different neuron, again, technically they don't touch, but where that area is would be called a synapse. So what occurs here with my pointer, and I'm pointing here, what occurs here? A synapse. What is a synapse? The connection, electrical connection between two different cells, meaning at least one neuron and another neuron, or one neuron and this could have been a muscle cell. One neuron and a gland. One neuron heart. Yeah, that is what allows the nerve impulse to leave from here and go on to the next cell. Otherwise, it would stop, and you would never get your end results. You've got to keep the impulse going.
You know how you said if it wasn't enough to pass through the axon hillock, it wouldn't continue to go. Okay, say it came from this one. Does it have to be as powerful to go through from that one to this through this one, or does the energy source start here?
No, it starts at the original source, which was the central nervous system. However, it might have just been one big long axon from the central nervous system, or it could have been a whole bunch of different ones, who knows, but that whole impulse would have started at the central nervous system to begin with.
Okay.
Now, whether at some point it may stop or it may not, but that depends upon the movement of particularly of sodium or potassium ions, which is much more detailed than we have to get into. Because actually what I'm saying is some signals are inhibitory.
Okay.
They'll let go to something and then tell it to stop.
Right.
You know, and that's when it would stop. Even though the central nervous system, that's where it originated at, yeah. It might actually go to something and tell it to stop doing what it's doing instead of keeping it going. But that was the purpose.
Right.
Okay, that's it for this.
At the end here, when this comes there, we have the telodendrite.
Yes, it will look just
So
like
the
this.
dendrite, the synaptic knob here, we have the telodendrite from this neuron plus the dendrite of a new neuron.
Yes, yep, and it will continue on. Yep.
You said a neuron can have more than one axon.
A neuron can have more than one.
Axon.
Oh, yeah. Sometimes there'll be a branch, like
At the end, right?
well, actually, it could be anywhere. That's where that one was at.
Okay.
But it might branch off right here, and that's the collateral. And it's just, and what it is an extra branch that many times is used for backup. Let's say so you had the collateral, it went this way. And then down the line, there was nerve injury, damage here that could repair. You might still if we can still stimulate that area with another part of that axon, you're all right. And it doesn't matter. So they're kind of backups or a way to get impulses to additional areas.
And you said the difference between peripheral and central nervous system, the neuron of the peripheral nervous system and that of the central nervous system
Yeah, that's how I can look at this model without anybody telling me or having it labeled, and I can tell you, because I mean, when I know it's by its shape, it's multipolar, which means it's either going to be motor neuron or it's going to be interneuron. So how do I figure out if it's in the central or peripheral nervous system? I looked at the myelin sheath. What does the myelin sheath look like? Is there one nucleus per myelin sheath segment? If yes, it's peripheral. Remember, if this were a oligodendrocyte, the nucleus is never attached. It'd be over here and have some foot extensions, but the main cell body would be over here. That would be central nervous system. Yeah. No. Well, the spinal cord, but you're not going to see this. They're microscopic inside the spinal cord. Yeah. Yeah. Okay, I got to call the next group up. Next group up, please make sure they get the first row. They get first row. You guys move around and behind them. Yes, don't hit my face. Okay, so everybody who didn't see is in first row? All right, do this again. Are you in the first row or second?
Yeah, first.
Huh?
The second.
Oh, you're first? Okay, so make sure she gets a spot. I'm going to start. Okay, so, second group. We have a multipolar neuron, which based upon the shape of its myelin sheath and the fact that there's one nucleus per section, I know that this is in the peripheral nervous system. So, it would be a motor neuron and a multipolar neuron. Let's start with these sections. These extensions are the dendrites. They receive signals. This entire area is the cell body. Inside the cell body, you have the nucleus. And around it, in blue, you have nisobodies. The nisobodies are similar to the endoplasmic reticulum, which is a series